Okay, students, this is chapter number 3, enzymes. In today's lesson, we will see what limiting factor is, and then we will see some factors which affect the rate of enzyme catalyzed reaction, or we can simply say the factors that affect the enzyme activity. These include temperature and pH. Then we will see the definition and consequences of denaturation. First, let's see what is limiting factor. Limiting factor is any factor that directly affects the rate of reaction. Varying the amount of limiting factor would affect the rate of reaction. Now, to understand these statements, let's consider these two graphs. If you see that in the first graph, I have taken substrate concentration on x-axis and rate of reaction on y-axis. Whenever we are taking anything or any variable on x-axis, it means that it is present in unlimited amount. We have ample amount of substrate. So now what is the limiting factor? Which thing is in limited quantity is the enzyme. Substrate is present in a lot of amount, a lot of quantity is there, but the quantity of enzyme is limited. So as we increase the concentration of enzyme and substrate, you see the reaction rate is increasing as well. But here you see that the rate of reaction has been gone constant. It's going constant from here because of the fact that since the enzyme was limited in amount, all the active site of the enzymes are being occupied till this point. Now substrate concentration, no matter how much it is present, this won't increase the rate of reaction unless and until we will add more enzyme, or the enzyme which are already being occupied would make the product and can be used again. But for a specific period of time, for a period of time here when all the active sites are being occupied, we say that the rate of reaction is now constant. Since the enzyme is limited in amount, the active sites were limited in amount, and all the active site of the enzyme are now being occupied with the substrate, or we can say all the enzymes are now saturated. They all are being occupied, so the rate of reaction is not increasing. In fact, it has gone constant. Now the rate of reaction would only increase, like I told you, if we will add more enzyme here, or the enzyme which are being occupied by the substrate would do the reaction, make the product, and can be used again in the reaction. Similarly, if you see here, I have taken concentration of enzyme on x-axis. Now what we have in unlimited amount is the enzyme. Now we don't have unlimited amount of substrate. Like in the previous graph, I have taken the rate of reaction on y-axis. Now what is happening, that rate of reaction is increasing as you are adding up substrate, but now after a fixed point of time or specific period of time, the rate of reaction is going constant. It is because of the fact that at this position the concentration of substrate was limited. So the whatever the quantity of substrate was present till this point is now being changed to product. Now the enzyme are there, but there is no substrate. The rate of reaction would increase only if I will add more substrate. So limiting factor in this graph was enzyme because it was lesser in amount, limited in amount, and adding it would increase the rate of reaction, or its absence has made the reaction constant. On the other hand, in this graph, if you see, the substrate is the limiting factor because the limited amount of substrate has made the reaction go constant, or you can say that only by adding the amount of substrate in the reaction we can increase the rate of reaction. So limiting factor can be an enzyme or a substrate depending upon whether they are in limited amount or not. So let me tell you the definition again. It's any factor that affects the rate of reaction and whose varying amount would affect the rate of reaction. Now come to the factors affecting the enzyme catalyzed reactions. First is temperature. I told you in our first lesson that enzymes are protein in nature and they are temperature sensitive. It means that they work on a specific temperature at their optimum level. Below the optimum level, they would be inactive, and if you will increase the temperature beyond the optimum temperature, the enzyme gets denatured. I will tell you the denaturation process in later slides. First, understand the effect of temperature. Enzymes are inactive at low temperature. As temperature is increased, the number of collision between the molecules, that is of substrate and enzyme, increases. More ES complex are formed and more product is formed. Hence, we say that the rate of reaction increases. The rate of reaction doubles with an increase in temperature of 10 Celsius. Understand these points by this graph. If you see that the graph is increasing as the temperature is increased, and here in the molecule section you can see that the rate of reaction is increasing because of the more collision between enzyme and substrate molecules. At low temperature they were not making as much product as they started to make at optimum temperature. But if you see further, after the optimum temperature, the rate of reaction is falling down. It is because of the fact that the structure of the enzyme is now destroyed. Since enzymes themselves are protein in nature, they are denatured at higher temperature. Different enzymes do work at different temperature, but most of the enzymes are not able to work at extreme temperature. For instance, enzymes of body, our human body, work at the temperature of around 37 Celsius. On the other hand, optimum temperature of enzyme papain found in papaya is about 65 Celsius. So this graph is very important, that you have to remember certain points. First point, that at low temperature enzymes are inactive since they have very less kinetic energy. As the temperature increases, the rate of collision between enzyme and substrate molecules start to increase, increasing the formation of enzyme substrate complex. Hence more product is formed. But as you will increase the temperature beyond optimum temperature, beyond the temperature where the enzymes were working at their best, if you would increase the temperature beyond that, the rate of reaction would start to decrease because of the distortion in the structure of enzyme. Beyond optimum temperature, the rate of reaction decreases as the three-dimensional structural change occur in enzyme. This is called denaturation. If you see the structure of enzyme, the protein structure is a globular structure, and when you increase the temperature, the bonds break, and the three-dimensional structural change, which is you can see in the diagram, is called denaturation. Here is another diagram to show you what denaturation is. It's actually a three-dimensional structural change in the enzyme caused by mild heat, acids and alkalis. Denaturation actually destroys the activity of enzyme by destroying the shape of the active site. Hence, no substrate can fit into the active site, no ES complex is formed, and no product is formed. Here you can see that normally the active site was allowing the substrate to fit into the active site, but as soon you increase the temperature beyond optimum temperature, the active site was destroyed, or you can say the enzyme was denatured, and now no substrate attached or fitted into the active site, no ES complex was formed, and hence no product was formed. Next is the activity and shape of enzyme, which is also affected by the pH. pH tells you the acidity or alkalinity of the medium, whether the solution or the medium is alkaline or acidic. You measure that by pH. Enzymes prefer to work at optimum pH. Outside of its pH range, the enzyme is denatured. So we got to know that every enzyme has a specific pH at which it works at its best. Here you can see I've given the example of pepsin and amylase. Pepsin is present in the stomach where the pH lies between 1 and 2. Here you can see the medium is acidic and the optimum pH for pepsin lies in this range. On the other hand, amylase work at around neutral pH, where you can see here in the diagram the optimum pH is lying at around 7. Amylase is found in pancreatic juice, as well as it is released by salivary glands, and the salivary amylase, which is released in the mouth. You know the substrate and product it forms. Amylase act on starch and convert it into maltose.